Dear friends, I will discuss some of the questions of previous years regarding nitrogen fixation in NET examination. So, first is this the material. These are the enzymes and their electron donor. For nitrogenase, electron donor is ferredoxin, reduced ferredoxin, and it has nitrogenase has Fe4, S4, and MOFE. Basically, structure of nitrogenase is very much like the nitrite reductase. Nitrite reductase has electron donor NADP and NADPH, and it has FAD, heme, tyrine, and tyrine is the molybdenum containing organic molecule. Nitrite reductase, ferredoxin is the electron donor. It has Fe4, S4, and heme. So heme is the part of nitrite reductase. Again, next question. Following statement were made with respect to the symbiotic association of rhizobia with legume. Note D is a regulatory gene. Note factors are lipochitin oligosaccharides. Note factors predominantly have a alpha 1,4 linked N-acetyl D-glucosamine backbone. So it is incorrect. Note C does not have alpha. It has beta 1,4 linked, this one. So receptors, lysine has the lysine domain is for receptors, correct. So answer should be fourth. A, B and D are correct. C is incorrect. Symbiotic nitrogen fixation in legumes and nodules involve complex interaction between rhizobium and legume roots. This complex interaction is governed by integration of symplasmid of rhizobia in root nuclear genome never happens. It never happens. So A is incorrect. Sensing of plants flavonoids by rhizobia, correct. Activation of node genes in rhizobia, again correct. Activation nodal genes in legume roots, again correct. So answer is the third one, B, C and D. The nodulation genes are classified as common node genes and host specific node genes. Some statement related to the such classification given below. Node A is a common node genes and node C is the host specific gene, incorrect, because A and C both are the node genes. Node B is a common node genes and node P is the host specific gene, correct. Node Q is common node genes, incorrect. Node H is a common node genes, incorrect. So answer should be the fourth one, that node B is a common node genes and node P is the host specific gene. So you can see previous material which I have given in this slide. Rhizobium gene that participate in legume nodules formation are called nodulation genes. The node D encoded proteins, it binds to the node box because it is the regulatory protein and induce transcription of all node genes. So second alternate is correct one. This is the match of the following involved in dinitrogen fixation with their respective genera. So you can solve easily. This question is very good question. Nostoc is phototropic free living. So D should be the third. Okay, and D should be the third in third and fourth. Now Frankia is heterotrophic nodulate, heterotrophic nodulate. Frankia is non-nodulate. So B second. So answer should be the third one. Given below are four statements related to agrobacterium mediated transfer of tDNA into plant cells. Production of single stranded tDNA by wire D1 and wire D2. This question was from December 18. Interaction of wire E2 with VIP1 and wire E3. Use of wire B1, wire D4 type of secretion system. Activation of wire A and wire G complex. The correct sequence of the events should be the fourth one. First activation of wire A and wire G, then the production of single stranded DNA, then the use of wire B that forms firecracker like structure, that is the channel for this one, and then interaction of wire E2, because wire E2 protects the single stranded T plasmid from host cytoplasmic enzyme. While designating experiment for agrobacterium mediated plant transformation, a student noted down the following points. TI-RI plasmid induced crown gall and hairy root disease respectively, correct. Enzymes octapine synthase and neopeline synthase involved in the synthesis of octapine and neopeline respectively, because neopeline is for the hairy root disease and octapine for crown gall disease. All the 6 wire genes, wire A, wire B, wire C, wire D, and wire E and wire G are absolutely required for virulence. No. Almost perfect 25 bases direct repeat sequence flanking all TI plasmid and RI plasmid in the tDNA is essential for tDNA transfer. So answer should be the fourth one, because all 6 wire genes are not involved, absolutely not involved, because either wire A or wire G alone start the activities. Electrons are transferred from reduced C enzymes like NADPH to pyruvic acid, its derivative during fermentation. So you have to match the product. So for this one, answer is the third one. Third one means streptomycin lactic acid, clostridium for butyric acid, and salmonella for succinic and acetic acid, and D this will the enterobacter for lactic acid. D for enterobacter for the butanol formic acid. Okay, so for tomorrow exam, good luck for all candidates. Wish you all the best and keep cool in filtration process, because you will see many questions. These are very difficult in first look, but if you have ability to filter the alternates, means if you know at least one correct question, correct alternate of that question, you can solve question that very easily. Okay, so good luck again.